In this module, we're going to be looking at using gene signatures and creating signature scores. To start with, we're going to be using the neuroblastoma dataset consisting of 88 tumors, and we're going to look at an analysis called the Kmeans or Kmeans clustering. So if you select under type of analysis and go down to meta-analysis, we're going to select Kmeans and click next. Here the correct dataset is already selected for us, so we can simply click select. And now you are presented with the adjustable settings in order to run the K-means analysis. In simple terms, K-means clustering is a clustering algorithm that is used to subdivide a dataset into a predefined number of groups. In this case, we're going to be dividing the dataset into two groups, and we're going to be dividing it based on the expression of a specific gene signature. So under gene filters, we're going to select our gene signature, and under gene sets, we're going to go in and select R2 provided gene lists, and then functional gene signatures. And under functional gene signatures, if you click one more time, you see here that you have a gene signature labeled IMR32 MIC-N all, consisting of 157 genes. These specific genes are derived from an experiment of the neuroblastoma cell line IMR32, which has had the deletion of MIC-N. So these are the 157 genes that were either up or down regulated following deletion of MIC-N in IMR32. So we're going to select this data set, and again leaving the number of groups of K-means clustering to 2, and the number of draws or number of repetitions of the clustering at 10 by 10. And we are going to click next. So you can see that we are presented then with a heat map, and at the top of the heat map you can see that there are subcategories that are listed as K-means. So the top one, which is labeled K-means, shows you which specific K-means cluster or group each sample or column is placed into. So you can see here that you have it divided into two groups, one group being the yellow group and one group being the purple group, and below that you can see the classification of each sample through each round of repetition of K-means. So you can see that there were certain samples that in certain rounds were classified as part of group 2. However, the overall clustering was defined into group 1 in this case for this specific sample. So what you can see is that through the different rounds, the majority of samples are clustered within the same cluster in each round, suggesting that these clusters that are defined through the K-means algorithm are quite stable. So if we then look down below, we have all the genes, the 157 genes that are contained within the gene signature. And you can see that they are somewhat divided into two different groups. You have one subgroup of the genes, which seems to be higher in the group 1 of the K-means, and you have another group that seems to be higher in the second group of samples. If you then look at what these samples actually are, what you can notice here is that if you look specifically at MIC-N amplification, you do have all of your MIC-N amplified tumors in group 2. What is interesting about this is that you also seem to have a number of other samples which are not MIC-N amplified, however they are clustering together with the MIC-N amplified tumors. So considering that the genes that are contained within this gene signature were derived from a MIC-N functional experiment, what this does suggest is that these specific samples that do fall into the same cluster as the MIC-N amplified tumors seem to be behaving in the same way as MIC-N amplified tumors, or have the same MIC-N signature, if you will, as these MIC-N amplified tumors. So this is one way that you can use K-means clustering in order to look into how specific gene signatures are represented in your data and how this can be used to then split your data up into subgroups. And by splitting your data up into subgroups, you are defining a categorical classification for each sample. So a group 1 versus group 2 in this case, based on the gene signature that you are using. Another approach to using gene signatures is that you might want to define a numerical or continuous measurement per sample. So that way you get an estimation of how strongly a gene signature is expressed in each sample. In order to do this, we're going to create signature scores. And in order to do a signature score, we're going to go back to the main page. We're going to again look into the same data set using the same gene signatures that we've looked into already, except this time we're going to go into meta-analysis and we're going to look at View Geneset or Heatmap under meta-analyses and click Next. So again, we're going to select the R2 provided gene list and we're going to click Next. And under the sub-collection of gene sets, we're going to select functional gene signature, click Next one more time. And here again, we are presented with a list of the different signatures that are available to us to select. So you'll notice here that under gene sets, we do have the gene set that has 157 genes, which is the one we looked at in the previous Kmeans analysis. And this consists of all the genes that were either up or down regulated in this McN experiment in the neuroblastoma cell line IMR32. What you can notice as well is that below it, from the same experiment, we have a subset that is labeled as DN or downregulated and a subset that is labeled as up. So this is the same 157 genes divided into two separate signatures that is downregulated and upregulated in the experiment. And we are going to select both of these two by simply holding Shift or Control while you select both two. And for the range of color scale in the heat map that we're going to view, I'm going to set that to 1 simply to intensify the differences between the groups that we look at in the heat map. And then we're going to click Next. We are now presented with our heat map of the two gene signatures that we've used, both the McN up and down. And what you can see here is that we have, again, a group of patients that are McN amplified, which seem to have high expression of a subset of genes, which if you look over here on the right, does correspond with genes that were included in the McN up signature. If you then scroll further down, you can see that there are a subset of genes amongst samples that are from the McN down signature, which again are higher expressed in the non-amplified tumors. Just the same as we saw in the Kmeans clustering, we do have a subset of tumors that are not McN amplified that do cluster together with the McN amplified tumors, as well as are high expressors of the subset of genes that are contained within the McN up regulated signature. So if we then scroll down to below the heat map, what you can then see here is that we have a Z score or signature score for each of the two gene signatures that we defined or used in this analysis. So you do get one for the up regulated signature as well as one for the down regulated signature, and this is essentially the Z score for each of those signatures for each sample. What you will also notice is that below both the up and the down, you do get something that is labeled W match. And now what the W match is that this is a weighted signature score that is essentially the up regulated signature minus the down regulated signature, which have been adjusted for the number of samples in each of these groups, or size adjusted. So this is the overall signature score of both the up and down regulated. And in order for this to be calculated when you do use one of the gene set analyses, you must have the same signature divided and labeled as both up and down or up and DN, which was in the case that we used here. So if we scroll down a little bit further, what you can then see is that you do have the option to save these signature scores, either as a text file, so the individual value for each sample, or you can store them in R2, which will then be stored as a track in your data, or as a pseudo gene that can then be used as a single continuous numerical value for each sample that we will then use for downstream analyses, and we'll show you how to do that. So we're going to click on Store in R2, and now we are going to store this signature as a track, and you can have the option of storing it either as a temporary track or as a personal track. So I'm going to, in this case, store it as a personal track, and I'm going to label it IMR32 McN weighted signature. And we are then going to click Build Set. So if we return to the heat map page, you can see that there was also the option to save each individual up or down signature. But now we have showed the example of saving the overall weighted signature, which we can then use as a proxy for McN activity within these samples. So in order to look at how we will use this in downstream analyses, we're going to return to the main page. And we're going to start by looking at a relate to track or a track by track comparison within the data using this MIC-N signature score that we have just saved. So we're going to click relate to tracks and click next. So here we are presented with the option of selecting the two tracks that we would like to relate to one another. So on the y-axis, we are going to now put our MIC-N weighted signature, which is now able to be found under the user-defined tracks. So I'm going to select that one for the y-axis. And on the x-axis, just to start off, we're going to look at MIC-N amplification, just to show that it is in fact related to MIC-N amplification, these samples. And we're going to view this as a box plot and click next. So here we are presented with the box plot of our signature score on the y-axis and MIC-N amplification on the x-axis. And here you can clearly see that the expression of your MIC-N signature score is much higher in those that are MIC-N amplified, and you have the associated p-value presented to you as well. So while this does serve as a positive control, that indeed we do see higher MIC activity in those that are MIC-N amplified, we can also correlate this with other tracks present in the data set. For instance, we might want to have a look at how it compares to the different stages of neuroblastoma disease, or the INSS classification. So we can click on that for x-axis and we can adjust the graph. And here again we can see that specifically in the stage 4 subset of tumors, which are the more aggressive tumors of neuroblastoma, that you do have a higher MIC-N activity or a higher expression of your signature score in this subgroup of patients. And this is, again, a significantly higher expression detected. So now we've looked at how this signature score can correlate to tracks that we have in our data. And we have seen that high expression of our signature score does correlate with high stage of the disease. So what we may now want to look at is how our signature score is related to overall survival or prognosis in these patients. So in order to do that, we're going to return to the main page. And we're going to make use of the Kaplan Meier analysis, which we have gone through in another module. So if we look at Kaplan Meier on the left, you can see that we can select both Kaplan Meier by track, which is a category, and numeric. And now our signature score itself is a numeric or continuous variable. So we're now going to select Kaplan Meier by track numeric. And we're going to scan our track, which means we will define the optimal cutoff automatically using the scan approach, and click next. And the track we are now going to select to use is our weighted MIC-N signature score. And we are now going to click next. So here we are presented with our Kaplan Meier plot, and you can see that high expression of our MIC-N signature score is associated with a poor prognosis. Now, interestingly, I have brought up a couple other plots here that we can compare to. So this is using our signature score, and you can see the associated statistical significance down here. If I then switch to looking at MIC-N amplification by itself, you do see a large separation between amplified and non-amplified with regard to overall survival. However, this separation is not as significant as using our signature score, and nor is looking at MIC-N expression itself. So it does seem that our signature score, which measures MIC-N activity, seems to be the best predictor of prognosis with regards to measuring MIC-N in neuroblastoma. So now we've looked at how we can make use of our signature score and how it correlates with different characteristics or tracks within our data, as well as overall survival. But what you might also want to know is, how does this signature correlate with other signatures or other pathways within your data set? So in order to look at this, we're going to return to the main page. And now we're going to select under meta-analysis, gene set versus gene set correlation. So we are now going to look at our specific MIC-N gene set and how it correlates to other gene sets. Another way that you could do this is by using track versus Geneset, in which case we could use our specific gene signature score to see how it correlates with other gene signatures. However, I'll describe to you why we're going to choose Geneset versus Geneset in this case in a little while. So we're going to set Geneset versus Geneset, click Next. And now as our input Geneset, we are going to select the R2 provided gene lists, and we are going to compare this to how the expression of hallmarks of cancer from the Broad Institute are expressed, and click Next. And as a subset of our gene signatures, we're going to select functional gene signatures and compare it to all the different Broad Institute hallmarks. And now you can see again that we have the option of selecting a subset of our gene signatures, and we are again going to select both the up and the down together in order for it to calculate a signature score the same way that we have done previously, and click Next. So because we have provided with both an up and down regulated gene list as an input, it does calculate the weighted signature score as we described before using the Geneset view. And this now correlates this weighted score with all of the different hallmark subsets that were available from this Broad Institute. And what you are provided here is you're provided with the Pearson R correlation between your signature score and signature scores from these other predefined signatures, as well as you are provided with information of the overlap between the different gene signatures. So here you can see that our gene signature had 149 unique, 3 overlapping with 53 unique to the target gene set, which would be MIC-N targets. So this, first of all, serves as a positive control because our MIC-N signature, in fact, does correlate most highly with a MIC-N or a MIC target signature with a Pearson value of 0.9. And you can also view these correlations in an XY plot by simply clicking on the XY plot. And here we are then presented with a 2 signature score plot and the correlating statistics with it. So if we then return to our gene set by gene set correlation list, you can also see that you do have some that are negatively correlated, for instance, and all of this information is provided for each individual gene set. Another interesting piece of information that you can extract from this is that while our gene set is positively associated with MIC targets, which is a positive control, you can also see that it does also correlate with E2F and G2M phase genes, which does then suggest that it is correlated with cellular proliferation and cell cycle. And again, the overlapping genes between the two signatures are quite small in all these cases, which means that the correlation that is detected is not due to the gene sets measuring the same genes. So in this module, we have looked at using predefined gene sets in order to create gene signatures and gene signature scores. And of course, R2 does allow you to create your own gene signatures and scores from these gene signatures, and that information will be provided in the Adapting R2 module.